Okay, I think we got a good number of people and it's about 1:05. So our first speaker today is Adithi Sundaresh from Helena Kilpinen's lab in Finland, who just changed into daylight savings time. So we're glad we coordinated that. I'm very excited to see what you have for us today. So I will stop sharing and you can jump on. Thanks so much for joining us.
Hi, everyone. My name is Adithi Sundaresh, and I am a PhD student in Helena Kilpinen's group at the University of Helsinki. So we're located in Helsinki in Finland, way, way, way up north. And today I'm really excited to be talking to you guys about our recent preprint that's titled Characterization of Cortical Neurodevelopment in vitro using gene expression and morphology profiles from single cells. So getting right into it, our lab, like many of you here in this audience, I think, we're interested in using iPSCs, human-induced pluripotent stem cells, to model neurodevelopment in a dish. And as many of us already know, iPSCs are an excellent model system for this kind of disorder because they allow us to model them with the right cellular, genetic, and temporal contexts. However, that being said, this particular piece of work, actually, we don't look at any kind of patient lines. We don't look at any specific disease. These are all lines from healthy donors. And the idea was more to set up a baseline, a characterization for the specific protocol, for the kind of cells that are produced, but also very importantly, to test out a different kind of assay and see how we could use that to assess heterogeneous cell systems. So we used 6 cell lines from 4 healthy donors, put them through this in vitro differentiation protocol from the Livesey group that was developed back in 2012. This is a 2D differentiation, so this is not organoids. For those of you who aren't from a neuroscience background, very quickly, the way it goes is it's a dual SMAD inhibition, small molecule-based approach. So kind of a gentle differentiation that's supposed to mimic in vivo development, starts from neuroepithelial cell patterning. These give rise to some neural progenitor cells, radioglia, which in turn undergo asymmetric divisions. These are really important cell types. They establish the progenitor pool, but they also give rise to these intermediate progenitors. And together, these generate a bunch of different kinds of neurons that form the different layers of the cortical plate. We sampled these at 3 time points in this piece of work. The first to correspond to the progenitors, the second to capture more radioglia, the intermediate progenitors, and maybe some early born neurons, and then the last one to capture the more maturing neurons. So very, very first thing was just to make sure that these time points are actually capturing what we want. And we did this with canonical markers by way of ICC. So just staining for them. And we found that, yeah, at day 20, we have some neural precursor cells. By day 40, there's some intermediate progenitors, early born cortical neurons as well. And then by day 70, there are the more maturing neurons as well as some astrocytes. Now, this is great, and morphology is indeed an important feature, especially with neuronal cultures, but we wanted kind of a bigger, more unbiased screen of the overall morphological features that would give us a broader overview of the differentiation. And this exists, for example, with the transcriptomic space, but when it comes to morphology, what we landed on was cell painting. And this is a really neat assay. It's a high-content imaging-based approach that essentially stains the different cellular organelles like mitochondria, endoplasmic reticulum, and then you can take images of these cells and extract all kinds of image-based features from them. And what I mean by that is for all of the different channels which correspond to the cellular compartments, we can extract features, things like the intensity of a channel, the texture, which is how the intensity varies over the image, but also we segment the individual cells in the images, and then we can assess the shape, the size, the density, and so on. And so this leads to really, really high throughput, high content data, and also can be obtained in high throughput and is quite cost effective. This assay has mostly been used in the realm of cancer studies for drug response testing, drug screening assays, basically. And this means 2 things. The first is that they're not generally looking at the features, the morphological features in terms of morphology, like really looking at the biology of it, but more using it as a way to partition cells in response to different drugs. And then the other thing is that those cell systems are far more homogeneous than our neuronal cultures. So there they generally apply the assay and then analyze it almost as a bulk screen. So they average all of these features that I just told you about across a well. And I mean like a physical well on the 384 well plate that this assay is usually done on. But instead, what we wanted to do was keep it at the single cell level because we thought that averaging it out across the well would basically average out all the biological meaning that we could get from this kind of system. So that's exactly what Dimitri, a postdoc from our group, did. He attempted to perform cellular phenotyping at single cell resolution. And essentially, with what I showed you before, where you segment the individual cells and then you get all of these image-based features per single cell, you end up with a high-dimensional matrix, very similar to what you would have with single-cell RNA sequencing data, which means we can do all of the same kinds of things. We can look at 2D representations of the dimensionally reduced data, and we can cluster the data. And this is exactly what I'm showing you here. On the left, well, on both sides, this is the UMAP from the cell painting data. So each point here is a cell, and on the left, these are just different examples of the cell painting features that are projected then onto the UMAP, things like the area, the density of the cells, or from the mitochondrial channel, the intensity or the texture. And then on the right is the same UMAP, but now annotated based on the clustering. And we actually went ahead and named these clusters based on what morphological features were the best markers of each of the clusters, as well as with the time point that was the most heavily represented in that cluster. And what you can see already by eye is that there's this trajectory that goes from the day 20 cells via the day 40 towards the day 70, kind of along that path. And another thing you can very quickly see is that the mitochondrial channel actually tags this trajectory very well. And this makes a lot of sense. We know that the mitochondria are extremely important in developing neurons. What I also wanted to show you was some representative images from each of these cell painting clusters. And you can see that they look extremely different from one another. And what I wanted to say with this is that these images, which by the way, these are what the stains are in the images, they really capture differences in cell biology, and hopefully the clusters are then also capturing these differences in cell biology. So this is what the neat thing is with this assay, that we can actually go back to looking at the cells that we're working with. And actually, we have an interactive web app that I can put a link into the chat later for those of you who are interested, that you can actually explore this UMAP and then start to see the images that are associated with those different points. But then kind of the big question became that we have all of these image-based features that sound kind of arbitrary, to be honest, and we wanted to know how can we assign biological context to these features. And so this is what we looked into, and we went to an assay that is most commonly used to assess cellular heterogeneity in vitro, and that is single-cell RNA sequencing. So we collected over 60,000 single cells from the same 3 time points, from the same differentiation protocol, from the same healthy donors, and we performed single-cell RNA sequencing. These are not the exact same cells, it's just the exact same experimental setup as the cell painting assay. And the very first thing we wanted to do was, of course, to annotate these cell types that we're producing. And we all know very well that in vitro is not the same as in vivo. But what we did was we used a reference mapping approach to try and annotate the cells based on an in vivo reference data set, rather than trying to do this with individual marker genes. And so we used the reference from Poliodakis, from cortical samples from gestation week 17 and 18. And what I'm showing now is a UMAP from the single cell transcriptomics data instead, and it's annotated by these reference mapped cell types. And we don't have to spend a lot of time on this other than to say that we do capture a lot of the heterogeneity that is seen in vivo, lots of different progenitor cell types, radioglia, intermediate progenitors, and then branching off into both excitatory and inhibitory neurons. And by constructing a pseudotime trajectory, then we also see that overall the developmental trajectory seems to follow what would be expected from what we know about in vivo. So all of this is great. It reassures us that we're producing relevant cell types, but then there's still the question of how to link this back to the cell painting assay. And so for this, we borrowed a method from this 2022 paper from the labs of Anne Carpenter and Shantanu Singh. And this basically is a method that takes advantage of the common experimental design between the cell painting and gene expression data sets to try and link the two via a predictive model. So our two data sets were linked by having common samples. And here by sample, I mean a specific cell line at a specific time point. And so we basically modeled the relationship between the gene expression and the cell painting data sets using a regression approach in which each cell painting feature can be estimated as a function of all of the genes in the transcriptomics assay. So this essentially gives us a coefficient matrix that links the two modalities shown, like as shown here. And the other thing that you need to know about the cell painting features is that they're highly modular, and there's often some redundancy in what they capture. So we grouped these into cell painting feature modules. So now by using this cell painting to gene expression coefficient matrix and these cell painting feature modules, we could put together a list of genes per cell painting feature module that was ranked by the gene's average contribution to the cell painting feature in the module. So you basically get this ranked gene list that is describing these morphological features. And using this, we can perform functional enrichment, which can then be used to annotate the clusters in a more interpretable way. So that's exactly what we did. So looks a bit complicated, this heat map, but it's really not. What you see here along the top are those same clusters that you saw in the cell painting heat map. And then here along the Y on the right in bold, you see the cell painting feature modules. And what's plotted is basically their activation scores. It's the same as if you were to plot a gene expression heat map, but we can't really call it expression. And these are basically the modules that are the best markers of each of these cell painting clusters. So on top of that, here we have this added layer of interpretability, right? So we have the top hits from the functional enrichment analysis, which can be seen as an annotation of what each of these cell painting feature modules are actually tagging. Now, of course, a smaller sample size limits these results from being generalizable, but at least in the context of this cellular model system, this adds an important layer of interpretability to the assay. And additionally, we also capture known or expected cellular biology, which is what really builds confidence in the approach. For example, you can see that intensity of mitochondria, the cell painting feature module, is associated with the mitochondrial ribosome as an annotation. And similarly, the endoplasmic reticulum is linked to the ER membrane. And so this makes a lot of sense. So now that we had a way of understanding the cell painting features, we wanted to move to a question that is important to our group and to many in this audience, and that is of donor-to-donor variation. It's really well known that cell lines can behave extremely differently from one another, and that any human-specific data set is also going to show a lot of intrinsic variability. And in our data set, we saw this donor-specific effect in inhibitory versus excitatory neuronal production, which we found to be especially interesting since interneurons have only recently been shown to be produced also from cortical progenitors, which is the case in our protocol. So what we actually can see here in this plot, it's an AMI plot, by the way. An AMI score basically it's adjusted mutual information, but it measures the dependence between the proportions of cell types and the donor of origin in this case. And what we can see is that at day 40, there are two donors that are really overproducing this MGE interneuron, an inhibitory cell type. And then by day 70, there's still one of the two donors that is consistently overproducing them. And we wanted to see if we could also see a similar effect from the cell painting data. And indeed, we found that the same donor from here in blue, it's overrepresented in one of the day 20 cell painting clusters and underrepresented consistently in a day 40 cluster. So we started to wonder, like, okay, how do we link this? How do we understand what these clusters are representing? So using this gene expression to a cell painting feature coefficient matrix, we performed a correlation in the space of marker genes of each of the cell types shown along the bottom here versus the predicted markers or the same marker genes via the predicted expression from the cell painting clusters. And we correlated the two to try and understand what cluster was representing what cell type. But unfortunately, what we found is that the cell painting assay, at least in this current form, is only able to capture broader neuronal cell types. And what I mean by that is you see this kind of broad clustering of the day 20 clusters from cell painting with more progenitor cell types, and then the day 40 and 70 with more neuronal cell types, but not a lot of resolution beyond that. So then to answer whether this is capturing a similar phenotype as what we saw from the single-cell RNA sequencing assay, we had to get a bit more creative. And we went back to the gene expression data, since this is more easily interpretable. And what I'm showing is a heat map of enrichment of selected KEGG pathways between the samples, because we wanted to see what kind of pathways were differentially enriched or depleted between the different donors. And these are ordered here by the proportion of inhibitory neurons that they were producing. And what we found was that the samples that are producing the most inhibitory neurons were consistently depleted in the ribosomal pathway. And this is actually interesting, because if you look at the clusters from cell painting, it's the same, this donor that's overproducing inhibitory neurons is upregulated in a cluster that's negative for endoplasmic reticulum, and concordantly, it is downregulated in a cluster that is positive for endoplasmic reticulum. And these are, I hope you follow along, but those are essentially the same thing. And then you also see this ribosomal effect. And so we dug a little bit more, and here I'm showing a donor that was more excitatory in its proportions and the donor that is more inhibitory, just comparing them. And we see that around the same point in pseudotime, so these are cells that are ordered in pseudotime, expression of a gene, and the colors are just the cell types. But anyway, you can see that the same time at which this transcription factor that's important for interneuron production starts to peak, you also see a drop in, I've shown an example here of one of the ribosomal genes, but this is also the case for others, which is not the case for the donor that is producing more of the excitatory neurons. And this is actually a known effect. It's been seen in literature that there are ribosomal differences between excitatory and inhibitory neurons, especially in mitochondrial ribosomes. But the point of the slide was not really to showcase like novel biological findings, but instead to demonstrate two things. The first, that donor-specific effects can indeed be picked up by the cell painting assay, which is interesting in itself. But then the second, that we can independently identify a plausible donor-specific phenotype in the two modalities in a way that's complementary to each other. And we found that really like the advantage of having this kind of a morphological screen. So now I want to take a small sidetrack from this main story to address an issue that I'm personally quite invested in, and this is one of cell type annotation, and I think this is a great audience to talk about this. Reference mapping, which is the approach that we use to annotate our cells, as a tool, as an approach, it annotates every single cell in your data set, no matter what. And what you actually need to do is then threshold for the quality of the mapping. And we found that a lot of published data out there doesn't necessarily do this. And when we did this, when we set a cutoff that the mapping score has to be at least 0.5, we found that about 40% of the cells across our data set fell under this cutoff, meaning that they were unmapped. They didn't really map to any one specific cell type from the reference. And at first we thought, okay, maybe there's just a cell type that's missing from the reference data set. But when we looked at the unmapped cells, they're kind of spread across the UMAP rather than being focally concentrated in any one region. And furthermore, although I don't show it here, they actually still express canonical markers of the cell type that they are closest to, but there's just still something different that's making their quality, let's say, lower than they should be to get mapped to a reference cell type. And we actually found that this has been a common problem also from some papers describing organoid cultures, that there are metabolic differences in vitro versus in vivo that can cause the cells to lack specificity to any one in vivo counterpart. And so we wanted to see if this was a similar case for us. And so what we did is we took this bucket of just these unmapped cells, and then we mapped those to a second reference, and this was an organoid reference. And what we found was that, first of all, we were able to map almost all of the unmapped cells. We were able to annotate almost all of the unmapped cells in this way, but also importantly, that most of them were then annotated as either this pan-radioglial or pan-neuronal cell type, which were cells that were described by the reference data set as being exactly this, these cells that can't undergo this bottleneck of subtype acquisition and remain with the kind of pan identity. They have some broader markers, but they're not quite what we would expect a cell type to be. And so I wanted to explore this a little bit more. And For example, with a pan-neuronal cell type, we found that across many different kinds of metabolic pathways, they had an intermediate activation to either the excitatory or the inhibitory neurons, and this could possibly lead to the inability to pass the bottleneck of subtype resolution. Now, overall, the main purpose of having this kind of a cellular model is to be able to apply it to disease research. And we wanted to know if the cell types that we're producing are actually relevant for this or not. So we use an approach called LD score regression, which is a statistical method that's used to understand genetic basis of complex traits, based on GWAS summary statistics. And it can be combined with the knowledge of gene expression to partition the heritability across different factors, in our case, cell types. So we basically used this stratified LD score regression based on GWAS summary statistics of many different common traits to try and test whether the disease heritability is enriched in certain cell type or tissue-specific genes. So here I've highlighted the results from a few key brain-related traits from our in vitro data set here and from GTEx's brain tissue on the right. And what you can see is that the cell types that we produce in vitro are indeed relevant to be able to study the brain-related traits. And of course, we can add some amount of fine-tuning resolution over the tissue level as expected. But the additional thing I wanted to point out is that in vitro specific cell types, the ones that were initially unmapped and then could be annotated based on organoid cell types, they're marked here with an O. There are some of them that are also found to be relevant. There are some examples here. And my point that I want to make is that these may not be perfect cell types, but they are still relevant for modeling the phenotypes that we're interested in. And my takeaway from this is that while we know without doubt that in vitro systems are not perfect, we can remind ourselves that most model systems are not, and still rest assured that at least they're able to capture relevant traits, which is important. So the story of that piece of work actually ends there, but I wanted to take a moment to mention some of the past and ongoing work in the group that I thought was relevant. Last fall, Pao, a postdoc from our group, published this article showing that somatic mutations can alter the differentiation outcomes of iPS-derived neurons. In essence, the findings showed that acquired mutations, especially in developmentally relevant genes, could influence the ability of a cell line to differentiate successfully. So a current working hypothesis in the group is that the metabolically restricted cells that I talked about just now might have acquired mutations that are preventing their successful differentiation. And this piece of analysis here, it depended on whole exome sequencing data for both the iPSCs and the parental fibroblasts from which they were obtained. And this kind of exome sequencing data is not always available and it's not always feasible to obtain. So we've been exploring alternative methods. And for example, there are some from the world of cancer research that can try and infer things like CNVs from single cell RNA sequencing data directly. And unfortunately, this one's a work in progress, but I would love to hear if you guys have opinions about this or ideas of how this could be taken forward, because mutational burden is certainly something that's important in IPS systems. So that's it. To quickly wrap up, I have hopefully shown you that cell painting can be applied to a heterogeneous cellular model system, can capture neuronal cell morphology, but to get even better resolution, we require some changes to this assay, and that's something we're trying to work on right now in the group, is tweaking the assay. We can also improve biological understanding of in vitro systems by complementing these kinds of modalities. That the cell types derived in vitro are certainly not perfect and require extensive characterization. That the in vitro systems still produce disease-relevant cell types. And in the future, amongst the many things that we want to work on, one thing is to have an increased sample size to look more into these donor-specific effects and this kind of cross-modality comparisons, and then to also, of course, apply this to model neurodevelopmental disorders. That's it. I want to thank everyone who was involved in this work, many people, but specifically Dimitri, who was my co-author on this, also Pao, Rina, Helena, of course, and Ziyu from Andrea Gana's group at FIM, who also helped out with the LD score analysis. And thank you for your patience, for listening. Happy to take questions.
Thank you. Questions? I think it's super cool that you're integrating some level of high throughput cell biology with high throughput gene expression. It's much easier for everybody in the world to measure the gene expression, but obviously getting to a more direct or just a very different view of what's happening in those cells that you're measuring gene expression is a very cool thing to be doing. Any other questions at this point? Maybe just a tiny quick technical one. I was very aware of what you were measuring in the imaging system, but not necessarily the details of how. Is it something that could be scalable so that it could be eventually coupled with a lot of this data in the future, this kind of imaging, or is it a fancier thing? I didn't catch all the components that went into that.
Coupled with this kind of data in what sense?
So if they say there's 100 single cell neural differentiation papers published in the next month, a year or two from now, how easy would it be for those groups to be incorporating some level of high throughput image analysis like you did as well? Is that?
Oh, I think pretty easy actually. It's well, the hardest part would be doing all the differentiations, I suppose, but you fix
Okay.
the cells and you dye them. They're these they're cellular dyes because they aren't antibody based, which is what makes them so cheap. The assay is very, very well put together. You can get a kit, you stain them, and you stick them in
Cool.
in an imager. So pretty
So
simple.
basically staining and then it's all image processing. Okay,
Yeah.
very cool. Sally, jump right in.
Yeah, that was so fantastic. Thank you for describing it. Very exciting to see how you put the cell painting on the UMAP and related. It's just awesome. So I have a question. I think you were doing 2D.
Yes.
Were you doing 2D cultures? So one thing I wanted to know was whether or not you'd transferred it to 3D in any way, number one. And then the second thing is you referred to donor differences, and I was wondering if to establish that they were donor differences, had you done multiple lines from the same donor, or are you referring to line-dependent differences?
To answer the first question, no, we haven't done anything in 3D. This is all work in 2D. I know that within the Institute, there's some efforts to try and put cell painting into application also for organoids, but it's not work that we are doing ourselves. For the second part, that's actually a very important clarification, yes. Although we have some clones from the same donor, different cell lines at an earlier time point. At the later time points, we don't. So it very well could be a cell line dependent thing. We're not entirely sure. At earlier stages, there are things that the different cell lines from the same donor certainly do correlate on, but we don't know if those translate into the effects that we're seeing later.
Thank you.